back. Well, I will show you the plates again. So we have, on the one hand, we have, this is how the plates are labeled. So let me do one thing. Okay, this is how the plates are labeled. So I means IPTG, right? You remember why we need the IPTG in the plate? Who can tell me why?
Ese induce el operón LAC.
Exactly. To have the lac operon on, right? And then we also, the X stands for XGAL. What is XGAL for? Who can tell me why we have XGAL?
XGAL is like the analog to lactose.
Exactly, is the analog that will be like the metabolite, right? That will be change of, like, that produces the change of color when it's metabolized by the enzyme, right? So we have, and then ARA, which is arabinose. So why did we need arabinose for?
To activate the plasmid with the
Homologous
repair sequence. Yeah.
Yes, exactly. And then, so we have one plate that has arabinose, and then the other one that doesn't have arabinose. It has only IPTG, XGAL, and the other one has IPTG, XGAL, and arabinose, okay? And we will finally have another plate, which is the one we will use today for the transformation. It has IPTG, it has XGAL, and it has SPT, which is streptomycin, which is the antibiotic for what is this antibiotic for?
To select the bacteria with our plasmid.
Exactly, to select the bacteria that will have the plasmid. Okay, do you notice something here? So we only have IPTG, XGAL, and streptomycin, but we don't have arabinose in this plate. Why? Okay, we don't have it because the CRISPR reaction will happen in the tube. So when we put the bacteria here, it will already be like happening. Is that clear? So here we do have arabinose, here the double strand break, and we already selected for those bacteria that have that system. We will transform them in the tube, and when we have everything in the tube, the CRISPR reaction will happen in the tube. And then we will put everything here. So there will be no need to select for the arabinose anymore, because the homology repair reaction will happen in the tube very fast. As when we add everything to the tube, everything will happen there, and then we will put it here, and we don't need arabinose anymore. Is that clear? Yes? No?
Yeah.
The homology repair we already like done before this plate,
Exactly.
right?
It happened
Okay.
in the tube. So here, what I have done is I have labeled tubes from A, B, C, D. Remember what we had in my slide? We will go back to that, but so I labeled these tubes. When we were asking what will happen in tube A, in tube B, in tube C, well, this is why I labeled them exactly like that. And I'm just going to do the transformation. Okay, so I'm gonna follow the protocol. It says label 4 tubes from A to D, which I already did. I added the transformation solution, which has calcium chloride. Did you remember why we need this for? We just talked about it before the break.
So the plasmatic membrane gets like more positive.
Yes, exactly. It's for the transformation. Okay, so this is what I have to do. Using an inoculation loop, pick 5 colonies from the IPTG X-gal plate and put them into tube A for at least 1 minute until all the bacteria are dispensed in the solution. And then immediately place the tube on ice. Okay, so I will just follow the protocol. I will take the bacteria from the XI, right, plate, and I will pick 5 with a loop. Let me bring okay, these are the inoculation loops that came with the kit, but I'm not going to use them because they are too big for my colony. So I'm going to bring one that I have from my lab that I think are a bit better. Okay, I'm gonna use this to pick only one bacteria, and I actually could use the other side. So I'm gonna take the plate, I put it on ice, and I'm going to pick 5 colonies, and I will put them into tube A. Okay, and then to tube B with another loop. So I'm gonna take one of these. Okay, I will pick 5 bacteria from the XI plate. Here the bacteria. I'm going to pick 5 colonies from this and put it in the tube A. Okay, for 1 minute. I think all the bacteria is now in the tube. And then place it on ice. And now I will take another 5 and put it in plate B. And try to pick from here. Put it into tube B. Okay. I think I have all the bacteria here. So the loop was blue a little bit, but now I got all the bacteria out and then it's not blue anymore. B. So remember, tube, maybe you can write it down, tube A and B. I'm putting bacteria that only has IPTG and X-gal, but no arabinose. And then tube C and tube D will have arabinose. So remember that for when you have to make the hypothesis of what's going to happen. Now I will take it from the arabinose. Our colony C and D. Okay. And the last one. D. Also, D is also with arabinose. I can still see some of the blue in the loop. Okay. Just by doing this, all the bacteria I pick from the plate is going to be transferred to the transformation solution. Okay. And this is how we are going to make the competent bacteria. Sometimes we can buy bacteria that is already competent, so we don't have to do this step. But this is not the case for this kit. Okay, place it on ice. So then it says, repeat step 3, repeat 5, okay. I did this. Okay, now what I'm going to do is I'm going to add the plasmid to the donor plasmid to tube A. So you should be writing it down so that then you know what to expect. So the donor plasmid will go to tube A. Okay, and then A and C, the donor plasmid will go to A and C. I have to add 10 microliters of the donor plasmid to A and C. So I take the 10 microliters pipette and I will place 10 microliters. From the donor plasmid to A and C. A. So we have donor plasmid here. We're just going to add. I do it like this so I can like mix it a little bit. Add it to A and C. Make sure it's the right plasmid, only donor plasmid. Then I will put it into A. And then I will take another tip and do it into C. Put it on ice, and then it says I should put the donor guide plasmid into tube B and D. So write that down. Donor plus guide into B and D. To B. And then to B. B and D. It's a little bit not completely tall, so I will do like this to mix it a little bit. It's still a little bit frozen, so have to do this and then incubate it on ice at least 10 minutes. And then we'll do the heat shock at 60 for 2 minutes. And we will then add the media. Very easy, as you can see. Hopefully it works though. I am doing it like this so I can mix it a little bit because it's still like you cannot see, but it has some ice. Okay. We add it to D. We leave it for 10 minutes. And I think we are almost done. Remember that here we are making the bacteria competent. So we'll leave it on ice. We'll put the timer for 10 minutes. We'll leave it on. And yeah, so I'm going to throw this. We'll wait for 10 minutes and then we will do the transformation. Inoculation loops. the plate. Going to wash my hands and we're almost done. Okay, we'll wait for the timer now. Okay, so it's like a cooking show. I feel like I'm cooking something. So we have to wait 10 minutes and we will then do the rest of the steps. And in the meantime, maybe we can like answer some of the questions that there are in the guide. Let's see. We have 10 minutes. Okay, so there are like, everyone please get into that link that I shared in the chat. And there are some pre-lab questions and some post-lab questions. You can we can go through them together. Oh, hold on, something happened to Soal. Her computer died and she left her charger in the lab and she cannot connect through her phone. Let me see. Are you guys from your phone?
No.
I was in my phone before and I could do it. Okay, so let's go over the lab questions. Everyone has their guide at hand? Yes. Okay. So I think we kind of cover some of the questions in class, but go to page the DNA focus questions, activity 2, page 11. Let me see where is Soad. So you will find an explanation of the you will have an explanation of the like experiment of what we talked about in class about non-homologous end joining and LAC, blue-white screen and stuff like that. So let's go over the questions in page 13. Do you see table 2? Page 13.
Yeah.
Okay, perfect. So we have the started plate conditions. Like I explained, IPTG, XGAL, color blue, Cas9, DNA repair off, and all of that. Okay. So let's go through the question 1A. So take a look at table 2, and then think about what we discussed, and then we will go over the question. Okay, 1 second. I'm talking to Soad. Okay, so it says, using evidence from table 2 explaining complete sentences why the bacteria colonies on the starter plates are blue. Entonces, ¿alguien puede explicar por qué? Porque ya lo hemos visto, pero para que esté súper claro, ¿por qué las bacterias son azules? Las bacterias con las que empezamos.
They have the donor, but they don't have the guide, so it won't cut out, and they will metabolize the X-gal, and metabolize its blue.
But where in the very beginning, like before we put the plasmids and all of that, the plates are blue, right? Like
yeah,
yeah,
because of the metabolite of X-gal.
Okay, so who can tell me what would happen if we didn't have IPTG? ¿Qué creen que pasaría si no hubiera IPTG? ¿De qué color serían las bacterias?
It would be white because the promoter wouldn't activate, so it wouldn't metabolize the sugars.
That's true. And what happens if we don't have X-gal? ¿De qué color serían las bacterias? Hay IPTG, pero no hay X-gal.
They would be white because the blue colors because of the metabolite.
They would be white because they don't have the metabolite, right? Is that clear for everyone? See?
Okay.
Yeah. Okay, so yes, well, we went through question B. If the bacteria on the started plates did not have a functional LAC-C gene, well, what color would you expect the colonies to be? ¿Qué color serían las bacterias? White, Anyone else wants to explain why? ¿Quién quiere explicar por qué serían blancas las bacterias? ¿No?
Porque no se podría metabolizar la X-gal.
Exacto. Okay. So that's, I think, very, very clear. Entonces, vamos con la pregunta C. ¿Cómo las diferencias entre los dos cultivos, uno es IPTG con X-gal y el otro que tiene arabinosa, van a influenciar el gene editing in the laboratory activity? ¿Quién puede explicarme eso?
If we cut the bacteria, the gene of the bacteria without the arabinosa, it is going to die because the arabinosa is responsible to activate the plasmid with the sequence of repairing, right?
Exactly. So with no arabinose, the promoter is not going to be active for the homology recombination, and then therefore the bacteria that have the guide and the Cas9 will die. You should maybe write down all of these. Okay, so let's go through page 14. We have these tables. So we will put everything together, and then maybe before we share it with the group, so that everyone has a time to think about it, go through try to fill in this table, what will be in each cup. traten de llenar esta tabla y luego como que comparten sus resultados. Mientras tanto voy a ver cuánto falta para... Nothing, almost 30 seconds. Ok. Entonces, voy a ver qué es lo que tenemos que hacer. 10 minutos. And then heat shock for 50 seconds, but it has to be 50 seconds sharp. Okay. I'm going to... Okay, the timer. Okay. I'm going to bring the computer closer to the water bath. And then I'm going to bring everything there and we'll do the transformation. Okay. Okay, so this is the water bath. It's at 60 degrees. I will bring the bacteria, so just hanging there. Okay. I will bring the protocol. Okay, so we will go through this. It says we did this part. We had 10 minutes on ice. Then we will put the bacteria 50 seconds exactly, 50 seconds, because if we go longer, the bacteria will die. And then we will place them on ice for 2 minutes. And after that, okay, using a pipette tip, we will add 250 microliters of LB media to each tube. And then we will leave them at room temperature for 20 minutes or overnight. Okay. Okay. Good. We can use this. I will use this to put the bacteria, the plasmids here and the bacteria, so that I can do 50 seconds for all the plasmids at the same time. So I will leave it, put the timer for 50 seconds exactly. Ah, 50 seconds. Okay, 50 seconds. En sus marcas, listos. Yeah. I'll show you how it's floating. So it will take 50 seconds. And after that, I have to put it in ice. Seis, 5, 4, 3, 2, 1. Okay, now I put it in ice for 2 minutes. Okay, we wait for 2 minutes and then I'm just going to add some media and we will wait for the bacteria to start growing for a little bit, like maybe like 20 minutes or so, and then we will put it into the plates, and then that's where we can see tomorrow the phenotype. Okay, so while we wait, we can go over the questions. I will bring the questionnaire. Okay. Okay, so go back to the questions. We were in page 14. Although we don't have much time, only 43 seconds. But so try to fill in the gaps of the table, okay? And then we'll discuss it. Take some time to read over and then see if you understood. Okay, I will add the media to the bacteria. This is LB media, it's called, which means LB broth, which is just like a lot of food for bacteria. So the protocol says we should let me see. Okay, says to add 250 microliters of LB nutrient broth to each tube. And then we close, we flick it, which means we mix it a little bit with the finger. And then we leave it at room temperature for 20 minutes or overnight. Okay, so I'm going to take some of these, 250. So this bottle has been autoclaved, which means it's sterile. Why do you think this is important? Who can tell me why we autoclaved it? Do you know what an autoclave is?
No.
No? I will show you. I will show you what it is. Okay, let's add 250. Okay. We'll add another 250. $250 more. And the last one. Okay, we will mix it like this. And we'll leave it at room temperature, which just means outside, for like 20 minutes or maybe, yeah, like before the class finishes, we will put them into the plate. Okay, last one. Okay, why do you think we're putting it at room temperature and not on ice anymore?
Because in this in this step, the cells are like recovering from the shock.
Yes, and then bacteria like to grow at 37, right? Or at room temperature. But when you put them in ice or in colder places, the bacteria actually won't grow that fast. It can grow, but it's not gonna grow very fast. Okay, so we will leave it so that it can recover and it can like divide a little bit, and then we will put the bacteria in the plate. An autoclave is a machine that we use. It's like a high pressure, almost like a high pressure cooking pot, I would say, that we use to sterilize things. So in this case, if we are using like something that the bacteria really like a culture media with a lot of sugar, I told you bacteria really like sugar. So that means that if we don't sterilize it, anything else can grow, right? It could have other types of bacteria that we don't want. We could have some fungus as well. We can have a lot of things growing there. So everything that we use has to be sterile. And usually when we are doing transformations or when we are working with plasmids and bacteria, we would work close to the flame so that we have like a sterile area to work. Okay, so last part, so that's it with the experiment. The last step is to put the bacteria like this in the plate. And then we'll leave it at 37 and tomorrow we'll see if it worked or not, okay? Let's see. Okay, we have the activity here. So I think you can like do this with all the information we have. I think you are able to do this by yourself because we have discussed it a little bit. So, okay, I think we can maybe go to the, go to our groups from last time and then continue with the bioinformatics part.